In this tutorial, we will explore cluster analysis using the Multiple Experiment Viewer. After downloading the software for Multiple Experiment Viewer, open the MEV application and load your expression data into the viewer. This is your file named significantvalues.txt. Search for your file and open. You may wish to load the annotations for Arabidopsis, which you can do under the Load Annotation Data box. Select Arabidopsis thaliana and the array platform, which is the ATH1 gene chip. After you load the data, click on the upper leftmost cell and then select Load. We now must normalize the color scale for the uploaded values. Click on Adjust Data, Gene Row Adjustments, Normalize Genes and Rows. Click on the original data and select Expression Image. Now we need to adjust our color scale limits. Click on Display, then Set Color Scale Limits. Next, fill in the boxes with the recommended values. For example, the top box recommended value is -2.27. Then click Update Limits and select OK. Next, we will perform cluster analysis using the Quality Threshold Clustering algorithm. This clustering method groups genes into high-quality clusters by setting limits on the cluster diameter to prevent dissimilar genes from being forced into the same cluster. Click Analysis, then Clustering, and select QTC. We will begin by using the default parameters in the box. These parameters determine the threshold for how similar expression profiles should be to be included in a cluster, and also the minimum number of gene expression profiles to include in each cluster. You can adjust these numbers to increase or decrease the stringency of your clustering. You can view the results by selecting QTC genes from the left column and then double-click Expression Graphs and select All Clusters. This provides an overview of expression patterns in the data. For example, Cluster 1 represents genes that are induced by nitrogen regardless of plant genotype. We can also explore individual clusters as heat maps by clicking on the expression image of interest. To save the clustering results, right-click on the All Cluster image in Expression Graphs and Save All Clusters. It is also important to save the MEV session file.